Welcome to the Whispers of Deep Time. I'm Alan, your host, and this is a podcast where science meets faith. Each podcast will tackle various topics from God to geology and life in the universe while searching for clues in our very existence. Join us as we take an objective view of the universe, searching for telltale signs of design and, most importantly, the designer. Before we go any further, and for full disclosure, we are people of faith, believers in a creator, and believers in science. But just because we are people of faith doesn't mean that we won't be objective. In our previous episode, we examined genetics and evolutionary mechanisms. Today, we go deeper to the origin of life itself, before natural selection, before replication, and before gene mutation. How did the first living systems arise from non-living chemistry? This field of investigation is called abiogenesis. But the deeper question is this: can undirected chemistry generate the kind of specified information and coordinated system that life requires? Intelligent systems arose that gave way to life and the abundance that we see today. This is where evolution falls short on explanations and become a big problem for most origin of life researchers. Maybe a bigger question is this: does the origin of life point towards something larger, intelligence? Today we will examine the scientific data and the logic carefully while searching for the telltale signs of intelligent design. Life is not merely complex chemistry. It is information rich, it's functionally integrated, it's self-replicating, it's energy processing, and it's compartmentalized. A minimal living cell requires genetic information storage, a translation system, metabolic pathways, membrane boundary structures. All of these components must work together for life to exist and replicate. Isolated chemical parts do not constitute life. From an engineering perspective, the origin of life is really a systems integration problem. But the question is, can chance bring about integration? In 1953, Stanley Miller and Harold Urey produced amino acids under simulated early Earth conditions. This demonstrated that organic molecules can form naturally. However, the atmosphere used was likely unrealistic. The experiment required artificial intervention. It produced amino acids, not proteins, and it did not produce information. Organic monomers are chemically simple relative to biological systems, but the leap from amino acids to functional protein networks is enormous and highly unlikely. The experiment showed plausibility of the building blocks forming unaided, but statistician William Dubinsky calculated the odds of this occurring at 10 to the power of 42,000 against this leap, or one chance in trillions. It didn't demonstrate the assembly of living systems. Chemical processes alone cannot explain the information barrier. DNA contains digitally encoded instructions. Its information structure is analogous to language or code created by intelligence. In all observed cases, specified functionally complex information arises from intelligence. We do not observe unguided chemical processes generating programming languages, symbolic systems, or instructional blueprints. Abiogenesis must explain the origin of functional nucleotide sequences, error correction mechanisms, and replication fidelity. Before replication exists, natural selection cannot operate. Selection requires self-replication. The first replicator cannot be explained by selection because it must exist prior to selection. The origin of functional information remains the central unsolved problem. The leading naturalistic model is the world RNA hypothesis. RNA can both store information and catalyze chemical reaction, but significant obstacles remain. Ribose synthesis under prebiotic conditions is unstable. Nucleotide assembly requires precise chemical conditions and information. RNA is highly susceptible to degradation in water. Self-replicating RNA systems have not been demonstrated under plausible early Earth conditions without intelligent laboratory intervention. Laboratory successes typically involve controlled environments, purified reagents, directed experimental design. These are intelligent inputs. They do not represent spontaneous geological or biological processes. Proteins require specific amino acid sequences to function. The combinatorial space of possible sequences is vast. Only a minute fraction yields stable functional folds. Random polymerization overwhelmingly produces non-functional chains. Even granting deep time and planetary scale chemistry, the search space problem remains significant. The question is not whether chemistry can generate complexity. It can, but lacks that functionality. The question is whether it can generate specified functional complexity without guidance or intelligent inputs. That distinction is central to the intelligent design theory. A minimal cell requires coordinated subsystems, information storage, translation machinery, energy production, and membrane regulation. These subsystems are interdependent. Remove one and the whole system collapses. This interdependence suggests threshold complexity. Incremental assembly must explain how partial systems confer survival advantage prior to full functionality. That explanation remains incomplete. Intelligent design does not argue, we don't understand it, therefore God. Rather, it argues when we observe high levels of specified functional integrated information and when unguided processes lack demonstrated capacity to generate it, inference to intelligent is reasonable and inference to a designer as well. We routinely infer intelligence in archaeology, cryptography, forensics, SETI research. When we see pattern signals beyond statistical noise, we infer a mind. DNA is not random polymer chemistry. It is information encoded in molecular form. The inference to intelligence is not religious by default. However, inference of a creator is evident. The inference to intelligence is based on uniform experience of cause and effect. If life bears signatures of intelligence, what does that imply? Well, it suggests the universe is not merely a self-organizing accident. Biological information may reflect intentionality. Chemistry may be the medium, but not the ultimate cause. This does not negate scientific investigation. Design detection is compatible with research. Recognizing intelligence does not halt inquiry. It is important to remain precise. Science has demonstrated organic chemistry is robust. Lipid membranes can form spontaneously. Catalytic RNA exists. Science has not demonstrated a complete unguided pathway from chemistry to a fully integrated self-replicating cell. Functionality in chemistry. That gap is not trivial. It concerns the origin of biological information itself. Abiogenesis remains one of the most profound unsolved problems of science. The evidence shows chemistry can generate building blocks, but integration and information remain unexplained in purely mechanistic terms. Whether that gap reflects temporary ignorance or a fundamental limitation of unguided processes is the central debate. Inference to design is a logical causal effect that cannot be ignored, given the knowledge of the specificity of life's cellular chemistry. Abiogenesis cannot as yet explain how evolutionary chemistry can create the necessary functional proteins in order to begin replication of cells. In this debate, presented with the competing hypotheses, we should select the one that makes the fewest assumptions and requires the least amount of unproven variables. Inference to intelligence is reasonable. Therefore, if inference to design is reasonable, then inference to a designer is also reasonable. In the next episode, we examine thermodynamics and entropy, and whether the laws of physics constrain evolutionary innovation. Until next time, this is Alan with Whispers of Deep Time.